Hey everybody, we're going to now talk about the chapter on the evolution of populations, or microevolution. I believe it's chapter 21. So a few learning goals that I have in mind for this chapter is I hope you'll be able to differentiate, and you should be able to do this one already, between the word gene, allele, genotype, and phenotype. You should be able to describe conditions that would prevent evolution from happening by the time this chapter is over. Give an allele or genotype frequency. If you're given one, you'll be able to use the Hardy-Weinberg equilibrium to see if evolution is occurring in a population for that gene. Predict how different types of natural selection would affect a model population, and given an example, predict why a population is evolving, flow, drift, etc. Those are just some examples of some of the things you should be able to do. So one thing I want to point out is that individuals, I've said this before, but individuals do not evolve. You do not evolve. You are born with a set of traits, a set of genetic traits, and those traits do not really change during your lifetime. You are a certain height, you have certain eye color, so on and so forth. Those traits really don't change during your lifetime. So you are born with a fixed set of traits. A fixed set of heritable traits anyway. So this finch is not evolving. But the population of finches can evolve. A population can evolve. An individual cannot evolve. Okay? So that's kind of an important concept to get down. Populations evolve over time. But individuals are stuck with what they got. Okay? Now, there was an experiment on the Galapagos Islands, your book talks about, before and after a drought. And they could see that the population, not just, there wasn't individuals that changed during this time, but the population changed over this couple-year period. So in 1976, the birds, on average, had a beak length of around 8.7 millimeters. After the drought, when food sources changed, the average beak length of the population was almost 10 millimeters. Now, birds did not grow longer beaks. The ones that had shorter beaks died off. The ones with longer beaks had more babies. The population changed over time. Okay, that's the important thing to note. Okay, so let's talk a bit about some Bio 1 level stuff as a reminder. Here we have two homologous chromosomes. A pair of homologous chromosomes. And here's the locus, that means the location of a gene. So remember, this bar represents a double helix of DNA, of paired bases, right? And right here is the location, the locus of a gene, in this case for flower color. And so it's the flower color gene, and there's, in this case, we'll just call that purple, and this one is white. Okay, so two different alleles. So we have a gene, which is a unit of heredity, in this case flower color, and then alleles, versions of that gene. And the way we're going to designate this, for our examples in this chapter, we are going to use, let me go back, we are going to use codominant alleles, something you'll learn more about in genetics class. But in this case, these are codominant alleles. They can both show up. There's not dominant and recessive. What I mean by that is we're going to use the letter C. If I can get my pen to work, let me jump back and forth for a second, see if I can get my pen to kick on. We're going to use the letter C to represent the allele. I'm sorry, represent the gene, color. And then we're going to use, in this case, P to designate purple, or W to designate white. Now, this leads to our phenotypes. So these are alleles, right? Purple color or white color gene. Now, genotype is in a diploid individual what two copies you have. So if you are CP, you're going to be have purple flowers. If you are CW, you have white flowers. Now, the reason we're drawing it this way, this codominant way, is because what happens when you have a heterozygous condition? If you are CW, now if this was regular Mendelian genetics like we did in Bio 1, if you were a purple and a white, you'd still be purple. correct? But in the case of codominant alleles, they both show up. So if you are CPCW, you might be a purple flower with white spots, okay? Something like that. Purple flower with white spots might be what you are. So they both show up. They're both dominant when they're both present, okay? So that's how we are going to designate genes and alleles in this chapter. So the genes will have a letter, and the allele will be kind of a superscript next to that letter, because they're at least when we deal with codominant alleles, that's how we'll do it. If we're dealing with straight up Mendelian genetics, you know, we can still do big P, big P, little p, right? Like we would have done in Bio 1. But with codominant alleles, you do it the way I've shown you. All right, so let's look at an example of a population. Here's a population of horses. And you can see some of them are red, some of them are white, and some of them are kind of mixtures of red and white. And I think there's some black horses in there as well. So you have all kinds of different colorations there. Variation. Populations vary in their phenotype. That's important to note. Populations have different phenotypes. And of course, that phenotype can be affected by evolution, assuming that phenotype is a heritable trait. So where does that variation come from? Genetic variation, well, first of all, it can be quantified as the average percent of loci, remember that means locations on a chromosome, that are heterozygous. So, you know, how much of the population is CPCW, purple and white? You know, what percent is CPCW? And you can quantify that in a population. Let's use the flower example in this case rather than the horse, right? Okay, that's one way you can look at it. What percent of the population is heterozygous for that trait? You can also look at the molecular level of DNA, and so nucleotide variability, which means, you know, if one critter is CGACAT, what's the other one? It might be GGACAA or something, right? So they can vary at that tiny nucleotide level as well. So it depends what you're measuring, how you might measure genetic variability. We're going to mostly stick to this big gene level, this allele level of variability here, the whole gene level, as they call it in your textbook. Nucleotide variation doesn't always result in phenotypic variation, because when nucleotides vary, it might not affect the organism, because a lot of your DNA are introns. This is a Bio 1 term that if you don't know, you'll have to look up. But a lot of your DNA is composed of, is what we call introns. It's non-coding DNA. So mutations there don't really affect the organism. But variations that occur in exons, they can affect the phenotype, but they don't always. They don't always affect the phenotype. Because if you remember, the genetic code is redundant. Several different codons can code for the same amino acid. So some phenotypic, some phenotypic variation does not result in genetic differences. That's something else we need to point out. Sometimes two organisms look different, but they have real similar genes, because the way we look can be influenced by the environment. For example, if you have the genes to be 10 feet tall, but you don't have a good diet when you're a kid, you won't be 10 feet tall, right? So the environment also affects your phenotype. So take-home messages here. Let me go back through these slides just to make sure we don't lose the thread. Populations are variable, and we can quantify that in two ways. We can look at how different the genes are in a population. So what's the proportion of the population that has heterozygous genes for a particular gene, CPCW. We could zoom in and look at the nucleotide differences. But we should keep in mind that nucleotide differences don't always result in changes in phenotype. It depends on where those mutations are, and even if they are in a gene, they don't always cause a change. They could be silent mutations, as you learned about in Bio 1. So lastly here on this slide, we need to remember that not all variation is caused by genes. Some variation is caused by the environment, but it's only genetic variation that can be affected by natural selection. That's the last bullet point here. Only variation that can be passed on to the next generation, heritable traits, are those that are affected by natural selection. All right. Now, just before we move on to the next video, I just wanted to show you an example of a gene here. So we can see here that genes can have lots of little mutations in lots of different places. And mutations that happen at introns aren't going to affect the phenotype very much, because introns are going to be cut out and removed, right, once we make our messenger RNA. Now, mutations inside the exons can affect the protein that will be produced. So deletions, substitutions, insertions, those may affect the phenotype. They may or may not. But I do want to point out that a lot of mutations are bad for an organism. Sometimes they don't have any effect. They might be silent. Other times they mess up the protein and they cause a problem. But rarely, but occasionally, they give the organism a slight benefit. And that's where the raw material of evolution comes from, where are these little mutations that cause new phenotypes to show up when we change the proteins that are produced from a gene.